Thank you for the kind introduction, and thank you for inviting me, Arthur, and the organizing committee. This is the first time I've been in Krakow, or first time in Poland, and we are enjoying it thoroughly. Fantastic hospitality, and got a glimpse of the history, and we are going to spend some more time here to really enjoy it. So I will talk to you about the treatment of relapse and refractory myeloma. If I can move to the next slide. Next slide, please. Thanks. So before I talk about individual drugs, I just wanted to let you know what we think about the biology, because at the end of it, you all will practice medicine as you want, as the drugs becomes available to you. So a couple of things I wanted to make sure or challenge. One of the idea how myeloma progresses is once the malignant clone is established as a monoclonal gammopathy, that over a period of time, this clone continues to multiply and then undergoes additional mutations and so on. And that way in MGUS to smoldering myeloma to multiple myeloma, and to plasma cell leukemia. This is only partially correct, not completely correct, because it also makes a question whether we should treat much earlier in the course of the disease in order to cure the myeloma. So here, what I wanted to show is another presentation that a newly diagnosed myeloma presents with, typically at this time, 4 to 6 clones. So this particular patient has, let's say, 5 different clones. But the drug-resistant clone, which is finally here blue, is just a sliver here, which was already present at the time of diagnosis even before you started treatment. So the patient did not develop treatment-induced mutation, and then finally you selected the drug-resistant clone. The drug-resistant clone was there in the beginning itself. If you had not treated this patient, we know from the studies done in New York in the 1950s, late 1950s, early 60s, the survival is 1 year. So why did now patients are living longer, 6 years, 7 years? Because you have introduced new drugs and you are able to bring the clone down eventually till the drug-resistant clone to the existing currently available drug emerges, and then the patient loses the battle to the cancer. So tomorrow if you have a new drug, and we will talk in the next slide, then you will again reverse it and the patient will live long. So that's an important concept to keep in mind, because people will say, I want to minimize the use of drug because I don't want to introduce drug resistance. Then there will be another group of physicians who will say, we want to put these drugs together because we want MRD. So there are two concepts here, and we have to understand the concept up front, that myeloma has multiple clones right from the beginning. So that's why combination therapy, triple therapy is better than doublet therapy, etc. Another thing I wanted to show is there are multiple mutations, but some of these mutations could also be targeted because drugs are approved for melanoma or for lung cancer, etc. They could be used for myeloma. If you know the predominant clone has the RAS mutation or something like that, then you can use a MEK inhibitor for that particular patient. So this one just gives you chronologically the progress which has happened, but you could see from 2000 onward, most of the progress has happened. Up until then, high-dose therapy and stem cell transplant was the king, and after that, now you can see that more drugs are coming and the future is changing. So in this particular slide, I just wanted to give you an idea about single-agent activity, and it's quite illustrated. When bortezomib was introduced, other than alkylating agent, there was no other drug in the field. So it gave you, as a single agent, 27% overall response rate, and duration of response was 12.5 months at that time for those who responded, almost a year. Then came lenalidomide as a single agent. Then comes carfilzomib. So when carfilzomib and pomalidomide was introduced, there was already bortezomib and lenalidomide in the clinic, or IMiD was already in the clinic, and still you got single agent activity which was robust, 24 and 18%. What is impressive now is when patients have been exposed to immunomodulatory molecules and proteasome inhibitor and autologous transplant and alkylating agent, single agent daratumumab was still able to produce 30% response rate and a duration of response of almost 8 months, showing that we are still able to bring novel therapies in this era by new mechanism of action. Then you can see the doublets given below and then triplets. Triplets are also illustrative because elotuzumab as a single agent was not very effective, but when combined with immunomodulatory molecule, elotuzumab was shown to be quite effective in prolonging the progression-free survival. And likewise, you could see ixazomib was also introduced in this particular era. But more important is HDAC inhibitor, single agent, no activity, but when combined with proteasome inhibitor, it also improved the outcome. So some of the drugs, new class of drug, could be introduced by combining with the existing drugs. Now, instead of giving you all the different studies which have been done, I just put them all in one slide just to illustrate that, again, 3 drugs are better. First one is Endeavor study. Carfilzomib is a better proteasome inhibitor. It could be dose escalated from 27 milligram per meter square, which was the initial approval, all the way up to 56 milligram per meter square twice weekly, or 70 milligram per meter square once a week. So the dose could be escalated, whereas bortezomib, you cannot escalate the dose. It causes death. So that's where the proteasome inhibitor carfilzomib is superior to bortezomib. The second one you can see is 3-drug ixazomib, lenalidomide, dexamethasone, or the third one, Eloquent, elotuzumab, lenalidomide, dexamethasone, then Aspire, carfilzomib, lenalidomide, dexamethasone. So 3 drugs are better than 2 drugs in relapse situation, and that should be kept in mind. And then you have recently daratumumab, which has been successfully combined with proteasome inhibitor or with immunomodulatory molecule, giving very good responses, something which we have not seen. So we will look at the results of CASTOR and POLLUX trial in the next 2 slides. So this is the POLLUX trial here. So you could see lenalidomide and dexamethasone versus daratumumab, lenalidomide and dexamethasone. And you could see that lenalidomide and dexamethasone is given continuously in both arms, but the daratumumab is continuous, and you can see there is a tremendous improvement in progression-free survival at about 18 months, 76 to 50%, almost 25% increase. And you can also see the depth of response is improved and the overall response rate is improved. Likewise, when you combine it with bortezomib and dexamethasone, again you have a dramatic responses at 12 months, 60% versus 22%. And you can see the overall response rate is better, the depth of response rate is better, just like the progression-free survival is much better. Now it has been combined with pomalidomide and dexamethasone and daratumumab, and you can see this 3-drug regimen is also exceptionally good, well tolerated, and you could see that you can achieve complete response and even MRD negative status. So this is important because we always worry about relapsed myeloma. Patient with myeloma, since in the audience there are patients with myeloma here, you should take to heart, sometimes your cancer may not have responded well to the drugs that was used up front, but then you come across a new drug and suddenly your cancer may be sensitive to this new drug, and therefore you can go into complete response and you can go into MRD negative status in a relapse situation. So these were all unheard of in the olden days when we had the same drug to recircle. Now these days we can see with the novel agent, we can even achieve minimal residue disease of deep responses, and these patients can do phenomenally well. So patients with relapsed myeloma can also get long survival, which was not heard of before. So relapsed patient, especially 1 to 3 relapses, their results are as good as what was once upon considered as a frontline therapy. So this is important to keep in mind. So what is the field happening? There are a number of new drugs coming up. So here you have two new immunomodulatory drugs which are coming in, CC122, CC220. Actually, there is a third drug coming up from that company, Celgene. So there will be three new immunomodulatory drugs going through the clinical trial. Then there is another HDAC inhibitor, which is HDAC6 inhibitor, better toxicity profile, and that's going through the clinical trial. There is a number of kinase inhibitor, which is going through clinical trial. Then you have novel mechanism, venetoclax and selinexor. So I will give you some data on those two new drugs. And then monoclonal antibodies, isatuximab, which is another anti-CD38 monoclonal antibody coming in. And then there are some checkpoint inhibitors. And more important is the bispecific antibodies and CAR T cells, which are coming into the clinical. So with all these progress, we expect, I would call this as a watershed period. If you can keep your myeloma patients alive for the next 3 years, possibly there will be a cure, perhaps with a CAR T cell. So it is important to keep in mind that this progress is a real progress. Then we can answer the question whether will there be auto transplant 5 years from now? I don't know. Maybe I will do induction followed by CAR T cell and that the patient is cured. We don't have to do auto transplant. So these are the progress which is coming into the field, which is remarkably good. So let's go through this one, which is nothing unusual. This is a different company with a different monoclonal antibody against CD38, and it is equally effective, could be given at a shorter time. The toxicity profile is very similar, and that's what is shown on this particular results. The data is shown here. The overall response rate is similar to what you see with daratumumab. Getting another company coming into the field only makes the drug more universally available and the cost comes down, so we all welcome that. This is a new drug, venetoclax. This is already approved for CLL. This is, works, it's an anti-apoptotic protein BCL2 and MCL1, promote multiple myeloma cell survival. Venetoclax precisely goes and binds to the BCL2 and therefore release the pro-apoptotic protein, so the myeloma cell will undergo cell death. And this is very important, and that's what you see here, that this is potentially effective. But in myeloma, BCL2 is not the key. In CLL it was the key, in some of the low-grade lymphoma it is the key, maybe in Waldenstrom it is the key, but not necessarily in myeloma. So the study was done in two different ways. One was study done by Shaji Kumar, and this was done as a monotherapy and with dexamethasone. And there Shaji noted that there is a group of myeloma patients who have 11-14 translocation. And in this particular patient, they quite often exhibit CD20 expression, more like lymphoma type phenotype, and these patients respond very well to this single agent. And you can see the overall response rate is up to 40% with this particular agent. Because this drug is already approved and in market in the United States, if I see 11-14 patient who's relapsed, I would be able to access the molecule and give it to my patient. So while the clinical trials are going on, having the drug in the market for another indication makes it available on a compassionate basis for individual patient you care about. So there was another trial in which they said we will block the BCL1 pathway, we'll block the BCL2 pathway, and we'll block the MCL1 pathway, we will block the BCL2 pathway. So that way myeloma patient will have a more robust response. And this was reported by Philip Moreau, and here they have treated 66 patients, and here you will see the overall response rate is much higher than the single agent results Shaji had reported, 67% response, because now not only the 11-14 patient responded, even the other patients who didn't have 11-14 translocation, they also responded to this. And again, high BCL2 patient had a much better response, but low BCL2 patient also responded because you had combined it with Velcade. Now comes a new drug, selinexor. It is a selective inhibitor of the exportin-1 protein. So what is an exportin-1 protein? So this is a shuttle protein which binds and is responsible for transporting protein from the nucleus to the cytoplasm. But what does this chaperone protein what are the cargo for the chaperone protein. So you see a number of tumor suppressor proteins which are all exported by this chaperone protein, exportin 1. And so if you block this, then these molecules, tumor suppressor proteins are not released, and then the cell undergoes apoptosis. So this would be universally applicable, not just for myeloma, it should be applicable for leukemia, solid tumors, etc. So this drug is actually explored in many different drugs, in AML, in myeloma, and so on and so forth. But I will just tell you the results in multiple myeloma patients. So here you have, the patients who had quad refractory and pentarefractory. So I'll just tell you what is the definition of quad refractory and pentarefractory in United States. So the patient should have failed the standard alkylating agent and the steroid. Patient should have also, if they were eligible for transplant, they should have failed transplant. They should have been exposed to 2 proteasome inhibitor, Velcade or bortezomib and carfilzomib. And they should have been failed 2 immunomodulatory molecule, lenalidomide and pomalidomide. So they'll become quad refractory. And they should have also failed daratumumab, in which case they'll become pentarefractory. So not only you would have failed alkylating agent, transplant, but you would have also failed the recently approved drugs. So in those patients, the patients were enrolled on this clinical trial, and you could see that the drug was quite effective. And the results are shown here actually by columns. So you have a quad refractory, and pentarefractory patient, and you can see the overall response rate is 20%. And if you kept in mind, as I told you, the bortezomib and carfilzomib and lenalidomide and pomalidomide, when they were approved as a single agent, their response rate was in the 20 to 30% range. So this is remarkable that this drug at the single agent would even show this kind of an activity in an advanced refractory myeloma patient. So then comes the checkpoint inhibitors and agonists. This is a slide just for myself so I can remember how this works. So there is a central checkpoint and then there is a peripheral checkpoint. So what I mean by central checkpoint is normally the T cells are educated by the antigen presenting cells. So naive T cells are educated to the antigen presented by the MHC, which you can see as a diamond, but in the color coding is poor, so you cannot see the HLA molecule, which is actually holding the antigen and educating the T cells. And the way you educate it with the co-stimulatory molecule and which binds there. But once the T cell gets activated, then CTLA4 is upregulated, then it could be turned off. So it could be activated. You can have a lot of T cells which are activated to an antigen, but it could also be turned off by getting the CTLA4. So if you use the drug ipilimumab or tremelimumab, 2 drugs approved by FDA, they are blocking the central education of the T cell. So you can educate a lot more T cells if you centrally block this particular agent. Then you have, peripherally, you have additional signals, CD70, CD27, navarilumab, which is also there. But now let's go to the peripheral, this thing. Now this is the myeloma cells. Myeloma cells turn off the activated C cells using the PDL1 or PDL2 mechanism. And now you can block this PD1, PDL1 by using nivolumab and pembrolizumab or pidilizumab, or PDL1 antibody, durvalumab and atezolizumab. So the atezolizumab is already also approved in the United States for bladder cell cancer. So you have a number of checkpoint inhibitors which are all approved for other cancers, especially for melanoma, lung cancer, bladder cell cancer, so on and so forth. See, the question is, what does it do in myeloma? So we had, Mateusz present this exciting data about lenalidomide dexamethasone when combined with the PD1 blocking agent pembrolizumab. They showed that they had tremendous efficacy, and the efficacy was good even in lenalidomide refractory patient. The response rate was 38%, and you even were able to achieve stringent complete remission and very good partial remission in 13% of the patient. So likewise, you have Budrose showing that it could be combined with pomalidomide and dexamethasone for advanced relapsed refractory myeloma, and again showing that the overall response rate is very good. So based on these data, then Merck immediately emerged and did two important clinical trials. One is Keynote 125, I can see the numbers there, not projecting well. So 165, which was a phase 3 randomized trial in relapsed and refractory myeloma, pomalidomide dexamethasone with or without pembrolizumab. This is the data which was just put out August 31, and if you go to that FDA website, it'll tell you now these drugs cannot be used for myeloma. So we had preliminary small data showing phenomenal effectivity, but yet now we are showing that the FDA put out an alert. You should not use checkpoint inhibitor with immunomodulatory molecule for myeloma. Why? Because you could show that the standard of care is in blue and the red is in pembrolizumab, and so you can see this is the progression-free survival. This is the overall survival, the first one, and then you have the progression-free survival. So they could not show an improvement in progression-free survival, but actually the overall survival was inferior because you could see grade 3-5 toxicity was much higher on the group of patients who received pembrolizumab with pomalidomide and dexamethasone. And serious adverse events requiring hospitalization was much higher if they received pembrolizumab. And these are all toxicities which we have not commonly encountered, and therefore we think it is related to pembrolizumab due to myocarditis, myocardial infarction, pericardial hemorrhage, cardiac failure, and so on and so forth. So sometimes the drugs may look very effective and the concept may be good, but we have to wait for clinical trials to really know whether it makes a big difference. This is for newly diagnosed myeloma, lenalidomide dexamethasone with pembrolizumab or PD-1. So newly diagnosed myeloma patients who were not eligible for transplant, when it was done again in this one, you have the overall survival. So the standard of care is blue, which is on the top. The investigational arm is in the bottom. So the survival was inferior, even in newly diagnosed myeloma patient, and there is not much of a difference. So again, you can see overall response rate was not much better, but the toxicity was much higher. Grade 3-5 toxicity was much higher. So this was interesting. I actually changed the slide just before coming to presentation because I had mailed in a slide which said that we want to use the checkpoint inhibitor in the future. It'll be great. Then after August 31 website, I had to change my slide and say, well, now this is not the way to go. But does this mean it is really the checkpoint inhibitors are dead? No, I don't think so. Sometimes the clinical trials gives you, when you do big studies, you can run into toxicity. Here is a patient of mine, newly diagnosed myeloma patient, who had high-risk complex karyotype with more than 5 abnormalities, had 414 and deletion TP53 or 17p deletion. This is very high-risk group of patients who have drug resistant. So the patient went on the triple regimen, 4 cycles, and we were all very happy, over 90% reduction in the paraprotein, but then during the stem cell harvest, the disease came back just during waiting for the harvest and collecting without chemo. Then you had Velcade Cytoxan Dex, but the disease actually started growing on that. So we used carfilzomib, pomalidomide, dexamethasone for 3 cycles, only slight reduction. We used a combination of VDSEP, only slight reduction. This patient, we did RNA sequencing. The MAP kinase, RAS MAP kinase pathway was highly active, so we used tremetinib and dabrafenib. So we had only a further reduction, but then the disease started growing up. So then we used the central and peripheral checkpoint inhibitor, ipilimumab and nivolumab for 2 cycles. Then the patient got autologous stem cell transplant, and then again got ipi-nivo, and then nivolumab and lenalidomide, which is what we said is, the FDA said was not the best. But this patient went into complete molecular remission, and the patient was just checked out a year later, still MRD negative complete remission. So the question is this because of transplant or is it actually due to immune manipulation? So what we did is we took blood samples ahead of time, and then we checked the blood sample for the presence of T cell clone. We realized that there were T cell clone. then we have a sophisticated computer program, because this patient's whole genome sequencing was done by us. So once you are able to do whole genome sequencing, you can use the program to find out what are the mutations, and next, which are the mutations which will be presented as antigen by the patient's own HLA. So once you define, then you find out whether the patient has T cell clone, which will recognize to the patient's own HLA presenting that neoantigen. Neoantigen means the mutation which was present in the patient's own myeloma cells. We found that even at the very beginning, there was a small amount of T cells which will proliferate to the neoantigen. But after the transplant, when we got the sample, you saw that those T cells expanded, and that is the reason we believe that the patient was able to go into complete remission, MRD negative. So this is 1 year out. If she's MRD negative next year, then we feel this is phenomenal. So we actually have a clinical trial now running with this approach. The next one is we have to talk about future. And this is about CAR T cell. So CAR means chimeric antigen receptor cell. And chimera is a Greek word, and the best way to tell it, especially everybody knows Indian god with an elephant head and a human body, Ganesh. So that's a chimera. So why is it called a chimera? Because normally T cells will only show T cell receptor. What we have is a B cell receptor, the variable region, snipped in, and it is expressed on the T cell along with the T cell signaling compartment. And this is done with the help of a virus, and which is what is shown here. So you take patient's own T cells, you virally transfect it and make it express the B cell, and in this case for myeloma it is against B cell maturation antigen or BCMA, and these T cells will become activated whenever they bind to the BCMA. So this CAR T cell clinical trials have been done, and we had a patient. For me, this was very illustrative. So there was a young gentleman, 36 years old, with a 6-year-old boy. So 36-year-old patient, he came with Bart Bolagi from Arkansas, because Bart Bolagi has come and joined me. Bart Bolagi has treated him in Arkansas for 4 years with 4 transplantation with all the chemotherapy and monoclonal antibody. The patient never went into complete remission, came to see Bolagi just to say goodbye, and he had 90% bone marrow involvement. At that time, we had a slot open for a CAR T cell. Bart Bolagi said, could you put this patient on the clinical trial? This patient was put on the clinical trial, and he went into the complete remission for the first time ever. And his blood count had not fully recovered after the CAR T cell. But the patient felt that I'm going home and I have to see my kid. And then he was going all over Europe with his son, because he himself doesn't believe he's going to live long, he's going to die, but this is one time he's in complete remission. And he's 6 months out, he's still in complete remission. So for me, that was eye-opening. So you could see these are the results which are presented there, that the patients who go into CAR T cell, who are at this time considered unmet medical need, they must have finished all the drugs available and go on the clinical trial. And within 1 month, the bone marrow, this is day 14 bone marrow, CAR T cell completely eliminates it. And there is a cytokine storm. It is a very toxic therapy. You should be ready to use appropriate treatment, tocilizumab, to rescue them. And you can see this is a PET CT. So we thought this was good. Oh my God. Then we had the results come from China, and they had a lot of it. And when I went to China, these hospitals now, they are investing on their hospital. They are 2,000-bed hospital. And their government's mandate is, we don't want to buy these American drugs. Each one is so expensive. Let's develop CAR T cell. So they have a CAR T cell program for many cancers. One of them is myeloma, and they're way ahead because they have treated. Now they are spinning it out as a company, and they are trying to come back to United States as a company to put out the CAR T cell. And they have shown that the patients who have gone into complete remission have stayed in complete remission more than a year. So this is really a new, exciting development. But CAR T cell is just now approved in the United States for treatment of childhood leukemia, CD19 CAR T cell. the last time I was looking for all these FDA thing, I just flipped to see how much it'll cost. It's about $400,000 to go through one CAR T cell for a child. So is that the solution? So we got to have some other solution. So there is another solution. We can get off-the-shelf monoclonal antibodies which will bind to the patient's T cells and to the patient's myeloma cells. So you will have one arm binding to the tumor cell and one arm binding to the patient's CD3 cells, bring them together and kill them. So this off-the-shelf antibody will be a lot cheaper, and even if the patient relapses, you can always give it back. And there are different ways of making antibodies. So this is a bispecific antibody. So there is a bispecific monoclonal antibody in myeloma from Janssen, first in human clinical trial, which will be happening in my center, hopefully in next month. And then there is another molecule which is called BITE. Here they only take out the variable region of the antibody from CD3, the variable region from CD19, but in our case it is BCMA. You join them together so the T cells and the tumor cell will come even closer, so it will be more effective. So there are other new technologies coming in. This is also a first in human clinical trial going to open up in our center very quickly. So these are all new trials which are coming. So the future is really exciting. So I always tell my patients, okay, if you're diagnosed with myeloma now, I can look you in the eye and say, you probably have a cure in your lifetime because of all these new treatments which are coming. So all I have to do today is keep you alive for the next 3 to 4 years. And I think with my experience, I can do that. 3 to 4 years, because the median life expectancy is so good. So I think really it is a nice time in myeloma with all the new drugs. But what I wanted to say is progress is coming because of new drugs and new therapies, and this is where we are. Thank you.
Professor Jagannath, I admired you and admire, and I cannot stop. Especially you are so flexible, you change your slides just before your lecture. I notice on many of your slides are already on the left side Delhi 2017 and on the right side Kraków 2017. You are already Krakowian. Thank you very much. You're very welcome. I also admire your data showing that there is additive or even synergistic effect between venetoclax and bortezomib, and it's fascinating because the different target. But coming back to resistant or refractory multiple myeloma, do you think that some cells develop different kind of resistance, different mechanism that they became resistant to the drug, or there is some kind of clonal selection that the cells which are a priori resistant dominate after sensitive cells disappeared with standard therapy?
Well, that's a very interesting question, and I put it there specifically because when you approach a patient and how you talk to the patient, this is a very important philosophical question. And this philosophy, I should say, came about thanks to Vincent Rajkumar and his MSMART put up on this. So they were saying that for good risk patient you use 2 drugs, and for high risk patient you use a 3 drug. This was a long time ago. Now they have changed the MSMART. But one of the philosophy is you don't want to use too many drugs up front, and you can always come up with another drug and rescue them. So that is one philosophy, and that philosophy only works if you believe that the treatment you gave rise to the resistance. But if you believe that even at the beginning there are multiple clones, now there is clear evidence from all the genomic studies coming from multiple centers that there are multiple clones even at the get-go. And that is why the results that 3 drugs are better than 2 comes out not only in relapsed myeloma, but we just now heard Turpo's talk about 3 drugs are better than 2, even in newly diagnosed myeloma. So then the question comes, can we add completely new drug, daratumumab, because that becomes an important question. So this is a philosophical question. I'm like you, I'm at the bedside taking care of patient. there are a lot of people in the labs who give one or the other answer. So that's something we should keep in mind, how we want to approach the patient personally. That's one. The second thing about the resistance, I think there are a subset of patients we talked about, like in the patient I treated who had 414 translocation, 17p deletion. Right from the get-go, that patient became very resistant just after 4 cycles of VRD. Then I could not capture a disease again. So that becomes a priority. Now we are doing RNA sequencing for these patients. And we are able to show, especially the highest risk patient, the 1416 translocation, the MAF. MAF is a transcription factor. And what does that transcription factor upregulate? It actually upregulates MYC. What does MYC do? And the MYC actually does 2 things. It upregulates the MAP kinase, RAS kinase pathway, and it also works on the BH3 molecule. So we have had patients who had failed multiple therapies, and we have used both the MEK inhibitor as well as venetoclax and proteasome inhibitor and HDAC inhibitor. And we found that the HDAC inhibitor with MEK inhibitor and tremetinib has put the patient into remission. So I think new insights in biology will come as we are understanding the RNA sequencing in the patient, individual patient. Now finally, single cell sequencing has come. Before, we always took bone marrow aspirate, purified at least 100,000 cells, and got their DNA or got their RNA and did an RNA sequence and got a heat map. That was population statistics. We used to say this patient looks like that patient or that patient looks like this patient, so they would behave. Now we can actually find out 4 or 5 different clones from each patient and see how each clone disappears or comes back as you give the treatment. This will revolutionize how we manage relapsed refractory myeloma. And finally, we will be able to be a clinician to choose which drug is the best for this patient, so-called personalized therapy. So I think the future is bright, but also it is conceivable. Hodgkin's disease, we cured MAP-ABV. This was even before I got into oncology, so you're talking about 1970s. Once Hodgkin disease was cured with 80% of the people going into complete remission, there was no progress in the field. The progress has now come now. We finally understood whether it is a B cell or a T cell, which is the actual cancer cell, what are the growth factors. Now we have a monoclonal antibody. Finally, we are bringing the monoclonal antibody for newly diagnosed Hodgkin's patients. So I do not know, while this robust progress in biology is going, I'm not sure where the CAR T cell is going to fit and how that is going to play. But my feeling is the biology is dramatically improving, whole genome sequencing, exon sequencing, but more important, I'm excited about single cell sequencing of large number of cells coming on board, and CyTOF, which is another technology where you can put up to 32 antibodies on a cell and precisely define the microenvironment. So you take the bone marrow aspirate and you can do CyTOF and actually see not only the tumor cell, but the surrounding immune composition. And you will be able to say why daratumumab was 40% in relapsed refractory as a single agent, 40% in newly diagnosed, 40% in smoldering myeloma. So even in smoldering myeloma, single agent daratumumab is not curing 80%. So there is something to it. Now we will be able to understand that. So I think the biology is exploding. Myeloma is a beautiful field to be at this time.
Thank you very much. Yes. Needs applause.